Welcome to the Deep Dive. Today, we're looking at something really central to India rice. The scale of our reliance on it is just immense, isn't it?
It absolutely is. Foundational for food security.
But here's the interesting bit. India actually has the biggest area globally for growing paddy, yet we're number 2 in production after China.
Right, and that largely boils down to yield. Our average output per hectare, well, it's lower.
Exactly. So let's unpack that because today's deep dive is about a really significant development that could potentially shift that picture quite a bit.
We're talking about the recent work by ICAR, the Indian Council of Agricultural Research. They've developed genome-edited rice varieties.
And this isn't just a minor tweak, right? This could have some pretty major implications for Indian farming yields, resilience, maybe even environmental aspect.
Precisely. It touches on a lot of key areas.
Yeah. And for you, the listener, especially if you're focusing on UPSC preparation, this is gold. It's core stuff for your daily UPSC mains current affairs, relevant for agricultural innovation questions in mains, and potentially giving you key facts for prelims too.
Consider this your sort of shortcut to getting up to speed on something important.
Yeah, think of it that way. We're drawing this mainly from recent reporting, including from the Indian Express, so it's current.
Okay, so where should we start? Maybe with the basics. What exactly is genome editing compared to, say, genetically modified or GM crops? People often mix these up.
Good starting point, because they are different. Genome editing, as I understand it, is about making really precise changes within the plant's own existing DNA.
That's the key. Think of it like using super accurate molecular scissors, technologies like CRISPR, to snip and edit the plant's own genetic code at very specific spots. You might delete a tiny bit or tweak something already there.
So refining the original blueprint, essentially.
Exactly. Now, GM crops, that's different. That involves inserting foreign DNA genes from a completely different species, maybe bacteria or another plant, into the target plant's genome.
Right, adding something entirely new from outside. Okay, that distinction is clear. So with that in mind, tell us about ICAR's big news here.
Well, ICAR has developed what are being called the world's first genome-edited rice varieties with these enhanced traits. There are 2 specific ones, DRR Dhan 100, which they're also calling Kamala, and Pusa DST Rice 1.
World firsts. Okay. And the key thing is these improvements better yields, more tolerance to stress. They got these without adding any foreign DNA.
That's the crucial point. It's all done by editing the rice's own genes.
Let's dig into Kamala first, DRR Dhan 100. Which part of ICAR worked on this one?
That was the ICAR Indian Institute of Rice Research down in Hyderabad.
Okay, IRR. And what was the starting point? The parent variety.
They use Samba Masuri, which is a really popular variety, well known for its grain and cooking quality. Farmers and consumers like it.
Right, and what specific gene did they target with the editing?
They went after a gene called cytokinin oxidase 2. Scientists sometimes call it CKX2 or GN1A. It's involved in grain development.
CKX2. And the technology used for the edit?
They use something called site-directed nuclease 1, or SDN1. This is one of those precise editing tools. It makes a targeted break in the DNA at the CKX2 gene site.
Like those molecular scissors you mentioned.
Exactly. And then the plant's own natural repair system fixes the break, but in a way that modifies the gene's function, slightly leading to the desired traits.
Okay, so what are those traits? What improvements does Kamala actually show?
The results are pretty impressive. First, it matures faster around 130 days. This is about 20 days earlier than the original Samba Masuri.
20 days earlier, that's significant. What else?
It also shows better drought tolerance. And importantly, it uses nitrogen fertilizer more efficiently. Less input needed, potentially.
Good for the environment and the farmer's wallet. And the yield, the bottom line.
That's the headline grabber. Average yield is around 5.37 tons per hectare, compared to about 4.5 for the parent. That's roughly a 19% jump.
19%. Wow.
And they say under optimal conditions, the potential could be even higher, maybe up to 9 tons per hectare. Crucially, it keeps the grain and cooking quality people like from Samba Masuri.
So it's not just more rice, it's still the good rice. That earlier maturity, does that have knock-on benefits?
Absolutely.
Huh.
It could free up land sooner for the next crop, or perhaps allow for different cropping patterns. Huge implications for farming systems.
Where is this variety likely to be grown? Which regions?
It's recommended for quite a wide area, agroclimatic zone 7, which includes states like Andhra, Telangana, Karnataka, Tamil Nadu, Kerala.
So large parts of the South.
Yes, and also Zone D Chhattisgarh, Maharashtra, Madhya Pradesh, even parts of Zone 3 like Odisha, Bihar, UP, West Bengal.
Okay, so broad potential reach. Let's switch to the other one. Pusa DST Rice 1. Who developed this?
This one comes from the ICAR Indian Agricultural Research Institute, IARI, in New Delhi.
IRI, okay. And the parent.
The parent variety here is MTU10LN.
And the target for editing. The name kind of gives it away. DST.
Exactly. They targeted the drought and salt tolerance, or DST, gene, again, using that SDN1 genome editing technique.
So the main benefits here are resilience to tough conditions, drought and salty soils.
Precisely. And the yield data under those stress conditions is quite striking. For instance, in inland salinity areas, they saw yields increase by almost 10%.
Okay.
On alkaline soils, it was nearly 15% higher. And in coastal saline conditions, it was over 30% higher yield compared to the original variety under those same stresses.
30% more yield in salty coastal areas. That's huge given the challenges there.
It really is. And again, the crucial point to reiterate, no foreign DNA. It's considered comparable to a conventionally bred variety from that perspective.
Which is important for regulations, which we'll get to. And the recommended zones for Pusa DST Rice 1.
Interestingly, same zones as Kamala. Zones of 7, Zone V, and parts of Zone 3. So again, wide applicability across major rice areas facing these stresses.
So we have these two varieties, higher yield, better resilience. If these get adopted on a large scale, what are the bigger picture benefits, beyond just the farm gate?
Well, let's imagine them being cultivated over, say, 5 million hectares, which isn't unrealistic given the target zones. The estimate suggests that could boost overall paddy production by about 4.5 million tons.
4.5 million tons more rice, significant for national food stocks.
Definitely. And then there's the environmental aspect. Kamala's shorter duration, needing maybe 3 fewer irrigation cycles.
Right, the water savings.
Exactly. That could save an estimated 7,500 million cubic meters of water nationally. That's water that could be used elsewhere or simply conserved. Plus, the better nitrogen use efficiency means potentially less fertilizer runoff and lower greenhouse gas emissions.
How much lower?
The estimate is a reduction of around 32,000 tons of greenhouse gases, about a 20% improvement associated with the nitrogen use.
Water savings, lower emissions, higher yield. It sounds quite promising. Now let's talk regulations. You stressed that these aren't GM. How does India regulate genome-edited crops like these?
This is a really key point, especially for policy watchers. India made a significant move relatively recently. The Environment Ministry issued guidelines that exempt genome-edited plants falling under SDN1 and SDN2 categories from the stringent GMO rules, specifically Rules 7 to 11 of the Environment Protection Act, 1986.
SDN1 and SDN2. So the types of edits used for Kamala and Pusa DST Rice 1?
Correct. SDN1 involves making cuts without adding foreign DNA. And SDN2 involves minor template-guided changes, again, without foreign genes, because they don't contain foreign DNA. They're regulated more like products of conventional breeding.
So a much simpler, faster path to approval compared to traditional GMOs.
Generally, yes. These two varieties, Kamala and Pusa DST Rice 1, already received clearance from the relevant biosafety committees, the Institutional Biosafety Committee, IBC, at ICAR, and the Review Committee on Genetic Manipulation, RCGM, back in May 2023. They were declared safe under this framework.
That streamlined approach must help get these innovations out to farmers faster. Are there any hiccups? What about intellectual property, IPR?
That's a valid question. There are always discussions around the IPR associated with the editing technologies themselves, like CRISPR. ICAR has acknowledged this and stated they are addressing these concerns to ensure things are handled appropriately.
Okay, something to watch, but it sounds like they're on it. Let's zoom out again. Why is this paddy breakthrough so important in the grand scheme of Indian agriculture? Remind us of the context.
Well, as we said at the start, paddy is just central. It's the main kharif crop, occupies a third of all our land under food grains, and accounts for something like 40% of our total food grain production. National food security hinges on it.
Indispensable.
Absolutely. And then you have that paradox we mentioned, largest area, but not the highest yield. Our national average rice yield is still below the world average, and significantly below countries like China, Indonesia, even Bangladesh sometimes.
So closing that yield gap is critical.
It's a major national priority. And innovations like these genome-edited varieties offer a powerful new tool to do just that, sustainably.
Is this push for genome editing happening just in rice, or is it broader?
Oh, it's much broader. There's active research using genome editing in other crucial crops, oilseeds, pulses, trying to improve their traits too.
Any examples?
Yes. For instance, the University of Delhi has developed a genome-edited mustard variety with improved oil quality and yield potential. And the government is backing this push.
How so?
There's been a specific allocation, I believe around 500 crore, earmarked specifically for genome editing research in agriculture. So there's clear policy and financial support to explore this technology across the board.
Right, a strategic push. Okay, let's try and synthesize this for our listeners, especially those with the UPSC lens on. What are the absolute key takeaways?
Okay, number one, ICAR has developed these two genome-edited rice varieties, Kamala and Pusa DST Rice 1. They offer higher yields, better climate resilience drought, salinity, and improved resource use efficiency like water and nitrogen.
Got it. Key varieties, key traits.
Number two, critically understand the difference. This is genome editing, SDN1, not traditional genetic modification involving foreign genes.
Distinction is crucial.
Number three, because of this distinction, they fall under a simplified regulatory pathway in India, exempt from the strictest GMO rules, allowing potentially faster adoption.
Simpler regulation.
And number four, the potential impact is huge, boosting national food security, helping farmers adapt to climate change, conserving resources like water, and reducing agriculture's environmental footprint. All major themes.
Absolutely. And for you, the listener preparing for UPSC, you can see how this connects everywhere. Agricultural technology, food security challenges, government policy, climate change adaptation, sustainable agriculture. It ticks all those boxes.
You could easily see Maine's questions touching on the role of biotechnology in agriculture or the challenges and opportunities of new breeding techniques. And the specifics, the variety names, the technology could pop up in prelims.
Definitely keep those details handy. So maybe a final thought to leave everyone with.
Well, it leads to the next question, doesn't it? We have this powerful technology showing promise. What else needs to happen? What further policy steps or research directions should India prioritize to really maximize the benefits of genome editing, while also making sure these advantages reach all farmers, big and small, equitably?
That's a great question to ponder. How do we translate the science into widespread, fair impact? Something to think about as you continue your UPSC preparation.
Indeed.
Okay, that brings us to the end of this deep dive. If you found this useful for your daily UPSC Maine's current affairs study, please do hit that like button, subscribe for more, and drop any questions you have in the comments below.
And remember, you can read more on this in the coverage by the Indian Express.
Thanks for joining us on the deep dive.